Hi, I am Dr. Arsalan and today we are going to discuss the Krebs cycle. Krebs cycle is one of the three stages of aerobic respiration or cellular respiration. The respiration has three stages. First one is glycolysis, in which the glucose is broken down into pyruvate molecules. Then pyruvate is oxidized into acetyl coenzyme A. Acetyl coenzyme A enters the Krebs cycle, the second phase of the aerobic respiration, and it is completely oxidized through these aforementioned steps in the Krebs cycle. Then there is electron transport chain as third stage of the respiration, in which the energy is released from the NADH and FADH2 molecules. So these three stages compose or compile the aerobic respiration. So first of all, we have to define the Krebs cycle. Krebs cycle is a metabolic or biochemical pathway which releases the energy stored in the acetyl coenzyme A by its complete oxidation through these steps. Krebs cycle was identified by Hans Wilhelm Krebs in 1937 and he got the Nobel Prize in 1953 for his achievement. Krebs cycle is also called citric acid cycle or tricarboxylic acid cycle. It is called citric acid cycle because of the product of its first reaction, the citrate or citric acid. To this molecule of citrate or citric acid, it is also called citric acid cycle. Krebs cycle is also known as tricarboxylic acid cycle because the citrate molecule has three carboxyl groups in its structural formula. Citrate has chemical formula of C6H8O7. The structure reveals that it has three carboxyl groups. That's why Krebs cycle is also called as tricarboxylic acid cycle or TCA cycle. So these were the synonyms of Krebs cycle. Krebs cycle produces the energy as well as NADH molecules as well as amino acids. So these three are the parts of Krebs cycle. Krebs cycle in eukaryotic cells, it takes place in mitochondria, while in prokaryotic cells or bacteria, it occurs in the cytosol of cell because of lack of mitochondria in the prokaryotes. As we know that the glucose molecule is converted into pyruvate by the glycolysis procedure. This pyruvate is then converted into acetyl coenzyme A, releasing the carbon dioxide molecule using pyruvate dehydrogenase complex enzyme. The pyruvate dehydrogenase complex enzyme is used to convert this pyruvate molecule into acetyl coenzyme A with the release of carbon dioxide molecule. The glucose molecule was 6 carbon molecule which was broken down by the process of glycolysis into two pyruvate molecules each of 3 carbons. Then again carbon dioxide was released, so leaving behind the acetyl coenzyme A that was 2 carbon molecule. This acetyl coenzyme A enters the Krebs cycle, which is completely oxidized in this Krebs cycle and release huge amount of energy. The Krebs cycle has 8 steps due to its 8 enzymes involved, which I have shown numerically that there are 8 enzymes involved in this Krebs cycle. So based upon these 8 enzymes, we can say the Krebs cycle has 8 steps in it. Most of the books they say that there are 9 or 10 steps in Krebs cycle by including the production of acetyl coenzyme A, but that was the separate component in which pyruvate was oxidized into acetyl coenzyme A, which cannot be included into the Krebs cycle. Therefore, the exact steps or enzymes involved in the Krebs cycle are 8 as clearly shown in this pictorial diagram. Now we will discuss each step in detail. First of all, the acetyl coenzyme A molecule coming from the pyruvate oxidation, as we have discussed, it combines with the oxaloacetate in the presence of water molecule in the reaction is mitochondria in eukaryotes and cytosol in the prokaryotes. Acetyl coenzyme A is 2 carbon molecule which combines to the oxaloacetate the 4 carbon molecule. So the resultant is citrate. Citrate is 6 carbon molecule. The enzyme involved or working here is citrate synthase because it is synthesizing the citrate molecule, therefore it's called citrate synthase. Then this citrate molecule is isomerized or converted into isocitrate in the process of aconitase enzyme. So it is also 6 carbon compound. Isocitrate is converted into alpha ketoglutarate by the release of a carbon dioxide molecule. So this 6 carbon compound will be converted into 5 carbon compound due to release of carbon dioxide molecule. Also, NAD is reduced into NADH in this step. NAD means nicotinamide adenine dinucleotide, while this molecule is called reduced nicotinamide adenine dinucleotide. I am writing here nicotinamide adenine dinucleotide. The enzyme used here is isocitrate dehydrogenase because reduction process is occurring here. Then alpha ketoglutarate molecule by the addition of coenzyme A is converted into succinyl coenzyme A. Again one carbon dioxide molecule is released here, so the 5 carbon compound will be converted into 4 carbon compound, and NAD is again reduced into NADH. The enzyme used here is alpha ketoglutarate dehydrogenase. The succinyl coenzyme A, it is converted into succinate. The number of carbons remains same onwardly in all the steps because of no gain or loss of carbon dioxide molecule. Succinyl coenzyme A is converted into succinate by the removal of coenzyme A molecule. Coenzyme A is removed and succinyl is converted into succinate. The enzyme used here is succinyl coenzyme A synthase. Along with it, a molecule of GDP, guanosine diphosphate, by the addition of phosphate molecule is converted into GTP, guanosine triphosphate, which is equivalent to ATP. Succinate molecule is converted into fumarate. The enzyme involved is succinate dehydrogenase. Again, this is 4 carbon compound, and FAD, flavin adenine dinucleotide, is reduced in this step. Reduced flavin adenine dinucleotide. Fumarate is converted into malate by the addition of water molecule. The enzyme fumarase is used here, and then malate is converted into oxaloacetate by the expense of malate dehydrogenase enzyme and reducing the NAD molecule into NADH. Again, the oxaloacetate molecule has been obtained because we had started from oxaloacetate and ended at the oxaloacetate. So this forms the cycle. Cycle is called the process in which the point of initiation and termination are same. We had started from the oxaloacetate and then ended at oxaloacetate, so it constitutes a cycle termed as Krebs cycle. A brief molecular review is the acetyl coenzyme A coming from the glycolysis pathway or pyruvate excitation combines with the oxaloacetate to form citrate, which is converted into isocitrate, then alpha ketoglutarate, then succinyl coenzyme A synthesized, converted into succinate, fumarate, malate, and then oxaloacetate. These are all 8 steps. At each step, an enzyme works for the conversion of next step. Now we will conclude the energy released during the process of Krebs cycle. First NADH molecule was obtained at this step, the second NAD molecule, and third NADH molecule. So NADH molecules obtained in the Krebs cycle were 3. One FADH2 was obtained and one GTP molecule was also synthesized. The NADH2 molecules, FADH2 and GTP, are converted into ATPs in next phase electron transport chain, which we will discuss in our next lecture. In electron transport chain, 3 ATPs are released from each NADH molecule, 2 ATPs are released from each FADH2 molecules, and GTP is converted into ATP molecule. Moreover, we should keep in mind that at the start of this lecture, I have told you that glucose was converted into 2 molecules of pyruvate because glucose was 6 carbon compound and pyruvate was 3 carbon compound. So 2 pyruvate molecules were obtained which were converted into 2 acetyl coenzyme A molecules. So in Krebs cycle, 2 molecules of acetyl coenzyme A were entering. Therefore, we will multiply this energy system by 2. So the NADH produced by one glucose molecule during the Krebs cycle will be 6, FADH2 produced will be 2, and GTP produced will be 2. Will be converted into ATPs in the electron transport chain. Directly, the ATPs are not synthesized in the Krebs cycle, but indirectly the Krebs cycle synthesized the molecules which have enormous amount of energy, and these are converted into ATPs in the electron transport chain. If we analyze the ATPs which will be produced from these products of Krebs cycle in the electron transport chain, then we can say that there are 6 NADH molecules and each NADH releases 3 ATPs. So 18 ATPs will be released from NADH molecules, and 2 molecules of FADH2 will release 4 ATPs because each FADH2 can convert it into 2 ATP molecules. GTP will produce 1 ATP, so there are 2 GTP molecules, so 2 ATPs will be synthesized here. So these will be net ATPs produced by the products of Krebs cycle. The sum will be 24 ATPs. So out of 36 ATPs of the glucose metabolism, 24 ATPs are contributed by the Krebs cycle by indirect way.